Okay, well, welcome to our second virtual class of this semester and this week. There's no Thomas. My children are still sleeping because they don't have school, so we'll see how this goes. But I do have my cat here. I'm gonna kick her out though, so you might hear her at some point. So here's some review questions for you from Wednesday. Um, so I'm gonna give you a minute to think about it. You can just pause the video and think about it, and then I'm gonna give you the answers. So, how are brown algae different from golden algae? Brown algae are multicellular. They have no yellow carotenoids. The golden algae are mostly unicellular and have both yellow and brown carotenoids. So, I'm sure that you said all that. Which protist causes red tides? I don't think we actually used the name, but it's Pfiesteria, which I should have put in italics, which is a dinoflagellate. Dinoflagellates cause red tides, and they also cause bioluminescence, and I fixed my video links, but now my iPad won't let me play the video links and share them for you. So I'll put those links for the bioluminescence and the swimming dinoflagellates, because remember they have those two flagella, the one that goes around and the one that sticks out, and that makes them spin while they swim. I'll put those links on Moodle. And then what's the unifying characteristic for the stromenopiles? It is they have a long hairy flagella, and then they also have a shorter smooth flagella, usually, but not always, because we're talking about protists. And then the last question, how are you planning to study for the test, and how have you been studying? That was just for you to think about and maybe talk to each other about. And if you come to our lecture, or you came to the lecture class, we talked about this some, and at the end of this PowerPoint, I'll talk a little bit more about what the test is going to look like. Okay, so we need to finish up talking about the alveolates, which is part of the SAR. The alveolates, the supergroup alveolates, we started talking about, and there were a couple things I wanted to make sure that we talked about, and my cat is really interested in these. So we talked about the stromenopiles, the diatoms, the golden algae, the brown algae, and alveolates. We talked about dinoflagellates, and then we need to talk about the apicomplexians, which I don't think I mentioned. I don't have a table in here, sorry. The and apicomplexians stands for apex, because it's at the end of the cell, and the complex stands for structures in the cell that help it enter host cells. So that's why they get their name apicomplexans. I need to move this bar so I can see my picture better. I know you can see. Okay. So we started going through the life cycle. We talked about the fact that most apicomplexans are parasites, that there's a sexual and asexual part of their life cycle, and they need two specific hosts. They need mosquitoes and people. So we talked about the sporozoites being in the saliva of the mosquito, which bites the person. They go to the liver cells, they infect the liver cells, they reproduce within the liver cells, and they are released as merozoites, which both of those are haploid. Those merozoites then infect red blood cells. The red blood cells, they're in there for that 48 to 72 hours, and that's when the person starts to feel like fever and chills. They're released as gametocytes, because they also reproduce in there. Some of those gametocytes then go off to infect other blood cells, and you can see this is just a picture of the merozoites inside the cell, the merozoite, and here's the apex, and that's where that complex is that lets it enter the red blood cell. I told you you'd probably see the cat. So then this is showing you red blood cells that are infected. This is not what our slide looked like in lecture. We did not find ones that had a lot in them, so I need to buy a new slide. So anyway, they're in the red blood cells. They're released as gametocytes, which are still haploid. Those gametocytes are in the blood, and when a mosquito bites again, that mosquito takes up the gametocytes, and once they're inside the body, the human body, they then, there's male and female, they develop into male and female gametes. Then they can fertilize. So the male, you can see, produces multiple gametes out of that one gametocyte, and the female only has one. There's fertilization occurs, After fertilization occurs, two haploid cells become a diploid cell. The zygote is a diploid, which develops this is happening in the digestive tract, the gut of the mosquito, and the zygote develops into an oocyst, which then explodes, releasing the sporocytes into the saliva of the mosquito, and this starts it all over again. So one thing that's hard about the Plasmodium is the name of the organism, and we'll use that word again later, not to be confused, is that they have the ability to, every generation, to change their surface markers of their cell. So they change the proteins that are the surface markers of their cell every generation. And so that makes it really hard for the body to fight it off, because your body responds to surface markers, antigens, that are on cells, and makes antibodies for that antigen. So if there's a new antigen every generation, every time the cells reproduce, they get new proteins. That makes it hard for the body to fight it off. So it's hard for the body to fight off because it can evade the immune system pretty easily. It's also hard to make a vaccine for that, because not only that, the cells inside the body are living, the apicomplexans are living inside cells in the body. So they live inside liver cells and red blood cells, so it's hard to even access them to treat them. So I was looking at the notes that I had on this from a couple of years ago, and it actually said that there's no effective vaccine for malaria, and we'll talk about that. But in October of 2021, so in the last 6 months, there was a a vaccine that was approved and recommended by the World Health Organization. It's a 4-injection series that's recommended to be given to children. And so they use part of this sporocyte protein, and that causes the body to make antibodies that prevent invasion of the liver cells. So when the body is able to respond to the sporocytes are what go into the liver. So if the body can respond to the sporocytes, that prevents the sporocytes from entering the liver. And so that kind of stops the life cycle early on in the process, so that you're not getting to the point where you're getting even symptoms like the fever and chills that happens when the apicomplexans have entered the red blood cell. And in addition to that, that also makes the body destroy, because there's antibodies against the sporocytes, it also makes the body destroy the liver cells that are infected. So it has kind of a two-part thing. What I read seemed to say it's about 70% effective. Some other ideas for combating malaria, because malaria has a lot of significant consequences. Countries that have high malarial infection rates have huge economic problems, have a lower economic, I shouldn't say huge economic problems, they have lower economic growth and development because people are sick. And you can get malaria over and over and over again because the surface proteins are changing in the apicomplexans, just like they do in the trypanosomes. It's hard to fight it off, so you can keep getting it. So another idea is to try to attack the mosquito population, and for a while that worked. They sprayed pesticides like even DDT, which has issues, but that brought down the numbers. In the 60s and 70s, the numbers were lower because of spraying pesticides for DDT and the development of drugs that were effective against malaria. But as time goes on, mosquitoes have become resistant to pesticides, and the medicines have become less effective, so there had to be new generations of medicines developed. But even now, if you were going to go visit a malaria-prone country, you would start taking anti-malarial drugs before you go. You take them while you're there, and you take them when you get back to try to prevent malaria. So, I think we're done talking about malaria now. And the apicomplexans. And we already did that. The last group of alveolates in the SAR group that we're talking about, the last clade, are the ciliates, which we looked at in lab, and some of you saw. If you did not see any paramecium, there is a video on Moodle that shows paramecium going around, swimming around. They're cute too. I think I say that about all of them. So ciliates, again, this video won't work, but ciliates have these lots and lots and lots of short little extensions. These are actually extensions of the cytoplasm all around the cell. They're like little flagella. They have microtubules in a 9+2 arrangement, just like the flagella are. And they, again, lots of them all over the body. They use the cilia to move, but they also use them to eat. So they move, but then they can also, these cilia will beat and they move water into this oral groove where the mouth is, so that they can bring food in. These organisms, and a lot of these organisms, live in hypotonic environments. So if you remember hypotonic, hypertonic, isotonic, hypotonic would mean that there are less solutes outside the cell than inside the cell, so water would move into the cell. And so they have to be able to deal with water continually coming in. So ciliates have a contractile vacuole, and the contractile vacuole helps them to control water levels. So it will take up water, and then every once in a while it will expel it out. So I think the video that I posted on Moodle for lab shows a contractile vacuole like filling up and then kind of spitting the water out. And they also have food vacuoles. So when food is brought in, it's put into a little vacuole. And they, you'll notice, have two nuclei. That's an important distinguishing characteristic of the ciliates. They have a micronucleus and a macronucleus. So the macronucleus is the one in charge of gene expression for everyday functions. So just normal gene expression, making proteins. They're also what we call polyploidy. My new plan for the stylus isn't working out. Polyploidy, meaning they have multiple copies of the genome. So haploid means one copy, diploid means two copies, which is normal, and for most things, for us. And then polyploidy just means multiple copies. Poly means many. So this is, again, this is normal gene expression. Then that micronucleus, the micronucleus is obviously smaller, but it's used in reproduction. And we're going to talk about the life cycle in a second, the process of conjugation, which is exchanging micronuclei for a form of sexual reproduction. And that's how they get genetic variation. So, again, on all these groups, you need to be thinking about, okay, what supergroup do they belong to? What clade do they belong to, like alveolates for ciliates, and what are their distinguishing characteristics? So ciliates have multiple small cilia. Some of them are all around their body. Sometimes it's in tufts. They have two nuclei. They have a contractile vacuole. That's it, basically. Okay, so here's our life cycle. So isn't this fun looking? Diploid and haploid are coming back up again. The word diploid, again, meaning two copies of the genome, and haploid meaning one copy. And you can see here it's color-coded, so anytime you see a green arrow, that means this is the process of conjugation. And then the blue arrow is asexual reproduction. So both of these are part of, and this is showing a paramecium. Okay, so we're going to start out up here. She likes. Okay, so we're starting with the compatible mates. So we have two compatible paramecium here, and the first thing that they're going to do is, if we start with that part, they're going to partially fuse. So here, there's a partial fusion. Again, notice that each of them has a we know that each of them has a macronucleus and a micronucleus. We're only showing the micronucleus here because that's what we're following, because that's what's involved in these processes. The process of conjugation and asexual reproduction is what's important. The micronucleus is what's important because that's what's involved here. So each of them also has a macronucleus. So we have a diploid micronucleus right now. These guys are going to fuse, but nothing's happening yet in terms of that. But the micronucleus itself is going to go through meiosis. So remember, meiosis takes a diploid cell, one diploid cell, and makes 4 haploid cells. So after meiosis now, these paramecium have 4 haploid micronuclei. nuclei. Each of them has 4. Okay? So they're partially fused, but no exchange of anything has happened. It's just the fusion triggers meiosis to happen. So now we have 4 haploid micronuclei. 3 of those are going to disintegrate. I'm trying to figure out why we have 5 here now. Maybe it is showing the micronucleus. 3 of those are going to disintegrate. No, I know what's happening, sorry. 3 of them are going to disintegrate, and the 1 is going to undergo mitosis. So we get rid of 3 of these, and the 1 that's left does mitosis, which copies it, so now there's 2. Okay, now each of them has 2 micronuclei, and they're going to exchange one of them. So the blue one's giving the purple one 1, and the purple one's giving the blue one 1. So now they each have 2 micronuclei. So that's the end of conjugation. At the end of conjugation, each paramecium has 2, or each ciliate has 2 micronuclei. So both of these are going to do this. I don't know why it's showing one of them going off. Then we get into asexual reproduction, which I guess maybe doesn't have to happen right away, and that's why that one's going off. They separate. The fusion breaks. When they separate, now these micronuclei are going to fuse. So we're taking 2 micronuclei, they're going to fuse and become 1 micronucleus, which is now diploid, because remember each of these was haploid, and n plus n makes 2n. So they are fusing. So we have a diploid micronucleus. That diploid micronucleus is going to go through 3 mitosis cycles. So it's dividing, going through 3 times so that we end up with 8 micronuclei. And again, these are genetically different than either of these 2 up here. They're different because of this fusion of the 2 different micronuclei from the 2 different cells. So now we have 8 micronuclei. 4 of these are going to become macronuclei. And the other 4 are just going to be, so the original macronucleus disintegrates, 4 of these micro ones become macro ones, and the cell divides twice. So we have 2 rounds of what's called binary fission, which is just an asexual reproduction and asexual splitting. So each one of these new cells gets one of those new micronuclei. So they each have a micronuclei, and they each get a macronuclei. And again, these macronuclei and these micronuclei are genetically different than these were. So this is a way for ciliates to have, to increase genetic diversity, to have genetic variation. Okay? Did you follow all that? So we have compatible mates. The compatible mates partly fuse. The diploid micronucleus undergoes meiosis. The haploid, then there are 4 haploid micronuclei. 3 of them disappear. The remaining one goes through mitosis, so now there's 2. Then they exchange them in conjugation. I'm pointing to this again. You can't see it. Then they separate, and we're only following this one. So these 2 haploid micronuclei will fuse to make 1 diploid micronucleus, which then undergoes mitosis to make 8 micronuclei. 4 of those become macronuclei, and the other 4 stay micronuclei, and the cell undergoes binary fission to make 4 cells that each have 1 macro and 1 micronucleus that are different than the originals. Okay. That is the end of the alveolates. So now we're going to move on to the last group of the last group in the SAR, the R of the SAR, which are the rhizarians, which, again, we looked at these in lecture. I need like a bigger lab here. We looked at these in lab. So rhizaria, okay, what are the unifying characteristics of rhizaria? Yeah. So they have a lot of different morphologies, so they look different physically. They include amoebas, but not all amoebas. They have, the main characteristic is they have pseudopodia. So they use pseudopodia to move and feed. So pseudopodia are just projections of the cytoplasm, so they can kind of stick the cytoplasm out, and the cytoplasm streams through that arm, and they move that way. So there's a video on the lab website, again, of amoeba, not this kind of amoeba, but amoebas that are in the unicont group, and how they move using cytoplasmic streaming and pseudopodia. We didn't really see that in lab. But not all amoebas are classified in the same group because, so an amoeba is an organism that uses pseudopodia, but because of other factors, they're not all in the same group. So there's some amoebas in this group, but when we use amoeba, we're generally talking about the amoebas that are uniconts. But like if I was generally saying amoeba, I would be talking about that. But these are technically amoebas as well. Most of them. So radiolarians we did not look at. Many of these are amoebas. Again, what makes them unique is that their pseudopodia, maybe I can keep writing this word over and over, are reinforced with microtubules inside them. So it makes them a little firmer. You can see these are pseudopodia. So they have microtubules inside their pseudopodia. So pseudopodia is just a cell membrane with cytoplasm in it, basically. But these guys have microtubules in them, so it makes them stronger. They also have a silica shell, like an internal skeleton. And they're kind of pretty, and I think I have another picture of one. Yes, but it's hard to see. So I'm going to take the pictures away again, and then we'll clear it at the end. Okay, then the foraminiferans, we did look at those in lab. They're sometimes also called forams, F-O-R-A-M. They, again, have a kind of shell. We call their shell a test. It's porous, so that the pseudopodia can actually protrude out of it. So they, that does not look like porous. The pseudopodia can come out through the holes in the test. And their test is made of calcium carbonate, which is like what chalk is made of. These guys are kind of cool because some of them look really cool. That's, first of all, some of them look cool, which hopefully you saw some cool ones in lab. They also can live symbiotically. With algae in their test, so they can have algae that are living in their shell, and those algae can help provide food for them through photosynthesis, and they help provide protection for the algae. So it's a mutualistic relationship, and then everybody's happy. Some of them are pretty large. They can be like several centimeters, and I'll show you a picture of that in a minute. And a lot of these are fossilized in sedimentary rock. So they can be found all throughout the world on land, fossilized in sedimentary rock. I gotta figure out, okay, yeah. So, let me erase all this, because now I can't see. would just recognize my finger, we'd be better off. Okay, it's better. Okay, so these are radiolarians, these top ones. And these are foraminiferans. The last group that falls into the rhizarians are the cercozoans. Cercozoans are another group that's just hard to talk about. There are some that are amoebas, and there are some that are flagellates. So there's some that have flagella. They don't have a lot of external, like, macro characteristics that unify them. Their unification, what makes them a group, is their molecular commonalities, which we're not going to talk about in detail. They do have thread-like pseudopodia, so really thin pseudopodia, unlike the ones that we'll see with the other amoebas. If they're cercozoans, thin pseudopodia are characteristic of them, and most of them they're heterotrophs, but some are parasites, and we're not going to talk about them much. We don't even have a picture of them. Okay, so that brings us to the end of SAR. So we've talked about the stromatophiles, the alveolates, and the rhizarians. Okay, the last groups that we're going to talk about are the archaeplastida and the uniconta groups. And you'll notice here, when we talk about archaeplastida, sorry, I had to push the button. When we talk about archaeplastida, we're talking about red algae and chlorophytes, which are green algae, and charophytes, which are green algae, but these are closely related to land plants. So again, we talk about how relationships change and our understanding changes. There's some people that argue that we should put all of these into one kingdom, because plants are in a different kingdom than these three. And so some people say we should put them all together because they're so closely related they should be in the same kingdom. But we're not there yet that I know of. But this changes. Uniconta, you'll notice, has some groups that we're talking about. We're going to talk about the amoebozoans and the episthecons, which includes things like slime molds, entamoebas, and those are the only two I think we're talking about. But you'll notice that also in this group are fungi and animals. So we're talking about things that are closely related enough that we're talking about us and animals and fungi, really large multicellular organisms that are closely enough related that they can be put into the same clade, the unicontic clade. So again, classification is interesting at times and almost always confusing, I think. So let's talk about the archaeplastids first. So we'll talk later about plants and fungi and animals, not a lot about plants. So plants, if you're a biology major, you'll take Bio 113, and we'll talk extensively about plants in there. But for the rest of you, if you're a biochem major or a biomedical sciences major and you only take Bio 111 and Bio 112, then we'll talk about fungi and animals and not really talk a lot about plants. So you should take 113 and talk about plants because plants are important too. Okay, so the archaeplastid supergroup is a group that we believe descended from a protist. That engulfed, I'm just going to write 8 because it's shorter, but engulfed is a better scientific word, a cyanobacteria. This is the idea of endosymbiosis, which your book talked about a little more extensively than we have in class. But that's the idea that organelles like chloroplasts came about because larger cells engulfed cyanobacteria to make chloroplasts, and my cat is writing on here. The mitochondria came around because larger cells engulfed heterotrophic cells, cells that were able to make energy using like cellular respiration processes. And so endosymbiosis is just the idea that that's the origin of chloroplasts and mitochondria, is from larger cells engulfing bacterial cells that then they kind of just started working together and became actual organelles. So again, archaeplastidas are close relatives to land plants, and we're going to talk about two groups. One of them are the red algae. So the red algae, you can see here are some pretty pictures of red algae. Red algae are characterized by having a specific pigment called phycoerythrin, which is red. It's oh, okay. So phycoerythrin is a red pigment. Chlorophyll is also present in these plants, but the red kind of masks it. But it depends on where it grows in the water. So red algae that's growing closer to the top is going to be more greenish red, and red algae that grows a little bit lower down is going to be more red, and red algae that grows really deep down is probably going to be like an almost dark reddish, maroonish, blackish kind of color. That I'm trying to figure out how to handle the cat. Okay, so these are also called rhodophytes. Rhodo meaning red. That color, the depth, the reason that having phycoerythrin is important is because they can absorb blue and green light. So chlorophyll, which is green, absorbs red and blue light the best. But phycoerythrin allows plants to absorb green light and blue light, and those colors of light, especially the blue light, can go even deeper. Blue light can penetrate water deeper, so that makes these plants adapted to be able to grow at deeper depths. They grow mostly in tropical oceans because they like warmer temperatures. They're multicellular. They can be filamentous, like you see here. They can be leafy. Again, we kind of use the word seaweed when we talk about them. The famous one, that's an N. The famous one is nori, which is the seaweed that's used to wrap sushi. And this is showing you a picture of how it's grown on nets to be spread into sheets. One thing that makes red algae unique from other algae is they do not have a flagellated stage. So in their life cycle, and the life cycles of algae are complicated. So they do not have a flagellated life stage, and that sets them apart from other algae like green algae, brown algae, golden algae. And some actually can be parasitic, which I thought was interesting. They've evolved to become heterotrophic and can be parasitic. The other algae, oh, I did make myself a new slide. Good job, me. Are the green algae. The green algae are called chlorophytes and charophytes. So the charophytes are the ones that are more similar to land plants. That's an A. So that makes them more similar to land plants, so we're not going to talk about them so much here. We're mainly focusing on the chlorophytes, which are green, just like both of them. Lots of chlorophyll in these, which is why they appear green. Again, very similar to land plants. They do have a biflagellated, so part of their life cycle, they have a 2 flagella. So they have biflagellated gametes, so that's in the sexual reproduction part of their life cycle. They can live pretty much anywhere. So some of them live in soil. We think of algae growing in water, but they can live in soil. They can have symbiotic relationships with organisms, and they can even grow in snow. So maybe there are green algae growing out there in the snow right now. They have a lot of different forms that they can take. So some of them, and these are 3 that we looked at in lab, Chlamydomonas are unicellular. So they're literally unicellular organisms. And you can see here there's 2 little flagella. I think they're cute too. And they can move on their own. Then you can get into some more complicated life arrangements. Spirogyra is an algae where it's multicellular. and it's become filamentous. And Volvox is colonial. So it's multicellular, a colonial algae. So they can start to have multicellular forms where they have different kinds of cells and differentiation, different kinds of jobs for the cells within that. Some of them actually have nuclear division, so the nucleus keeps dividing, but they don't divide the cytoplasm, so they end up being these multinucleated cells. We don't have a picture of one of those. But again, they have a flagellated they have flagellated gametes, they have a lot of chlorophyll, and they have a lot of various life forms all the way from unicellular to multicellular with differentiation in them. Okay, so that's Archaeplastida. And last but not least, we're going to talk about the uniconts. Oh, a picture. Oh, no, that's so this is the last supergroup we're talking about, the amoebozoans. Again, this is a really hard group to define. It's a pretty recent group that has been formed. It's not really related to other supergroups, and it's just very diverse. That's its unifying characteristic. It's very diverse. Because it includes things like fungi, it includes things like animals, it includes slime molds and amoebas. So it's a really odd group. There's two major clades in it, the amoebozoans and the episthecons, that have a very loose connection. So we're going to talk about episthecons in a little bit. Did I take all those pictures out? I'm sorry. No, I put them on the next slide. So amoebozoans, they're amoebozoans, so they're amoebas, so they have pseudopodia. And their pseudopodia are bigger. They're kind of tube-like. These are pseudopods. And again, the cell is changing shape and moving the cytoplasm and changing its membrane to ooze its way to other places. So they use those, sorry, for feeding and for movement. This includes the amoeba protists, which is what we looked at in lab. There are some really rare amoebas, thankfully rare amoebas, that can be found in aquatic environments that cause dysentery and can eat your brain. So every once in a while you'll hear about someone who is swimming in a body of water and got this really weird illness where their brain was breaking down because an amoeba got into their brain. This group also includes slime molds, which is what we were going to do in lab, which is sad. Slime molds, there are two different groups of slime molds. There are the plasmodial slime molds, and that's what we were going to work with, and hopefully will work with, although they're supposed to arrive today. These are in this group because they have amoeboid feeding stages. No, I don't want to say that. The plasmodium do. They have an amoeboid form. So all slime molds have an amoeboid form. The plasmodial ones have an amoeboid feeding one. They also form fruiting bodies. And those fruiting bodies are what produce the spores for reproduction and their life cycle. So the plasmodium ones are really brightly colored. They're often like orange or yellow. Ours will be yellow. And then the feeding stage has multinucleated. So we have a multinucleated feeding stage, which we call a supercell. And it kind of oozes around like an amoeba. It has cytoplasmic streaming, and sometimes you can actually even see it under the microscope. You can see the cytoplasm streaming, and it oozes around to find food. This supercell is called a plasmodium, which is confusing because plasmodium is the organism that causes malaria. but it is not malaria. When food runs out, so when there's no food, these like moist environments, so if there's a drought or in their environment that's drying out, so these are land organisms, they will kind of, you might see them on like rotting logs or under leaves or something like that. When they dry out, when conditions get bad, when there's no food, they form spores. And those spores can withstand drought conditions and stuff like that, and then they can regenerate into the plasmodium again when conditions are good. And again, it's the fruiting bodies that are making spores. The other group of slime molds are the cellular slime molds, which are not quite as fun. Their feeding stage has only one nucleus. It's a single nucleated cell. But when food is running out, they will actually combine. They will group together. They will aggregate to form a multinucleate. Multicellular, I want to say. A multicellular group, body, and that will then make a fruiting body to make spores. And this, I move these pictures. These are pictures of these things. So these are fruiting bodies. This is a plasmodium. So it's a plasmodial slime mold. So you can see it's bright yellow and it's got these weird forms. Again, cytoplasm streaming through that helps it move. These are the fruiting bodies that are going to produce spores. And this is the fruiting body. This is the multicellular aggregate and the fruiting body of a cellular slime mold. So that is the end of our classification stuff. We have a couple more things to talk about with, oops, we're not talking about opisthecons now. We'll talk more about that later. Last thing we're going to talk about is the role of protists in the environment. You know that we've talked about protists that make you sick, but protists are really important in the environment. And we're going to talk about kind of two major roles that they have in the environment. One is symbiosis. And this can be in a good way through mutualism or commensalism, which is neutral, or in a bad way through parasitism. There's a couple pictures here of coral reefs showing you. Coral reefs are a symbiosis of dinoflagellates and corals. So the dinoflagellates live inside the coral. They do photosynthesis to bring in nutrients for the coral, and again, the coral offers it some kind of protection. It's not just dinoflagellates and corals. There's also bacteria and other things that are involved with that. So corals are really, the organism coral is really a community of organisms. The problem is when corals get stressed or when conditions happen, the corals can actually release the dinoflagellates. They eject them out, and that can be temporary or it can be more long-term. And this can lead to what we see here, which is coral bleaching, where the coral doesn't have any pigments, it doesn't have color, and it can't get the nutrients that it needs, and it can end up dying from that. So the polyps die, the coral polyps, which we haven't talked about their life structure yet, but the coral polyps die without the dinoflagellates long term, and without all their symbionts long term, and so then you're left with this coral skeleton of dead coral, which is hard calcium carbonate. So an important role in that. They also are involved in digesting wood. This is a picture of the protists that live in the gut of the termites that helps them digest cellulose. That allows the termites then to absorb the material. So is this a good symbiotic relationship for the termite? Yes. For humans, no. But it's an important one that has a lot of economic impact to the life of termites. And again, pathogens. We talked about some different pathogens like malaria, trichomonas. There are also ones that affect plants that are protists. Sudden oak death is caused by a protist. So lots of different roles in symbiosis. Again, some positive and some negative in terms of our point of view at least. The other really important role that a lot of protists have in the world is photosynthesis. Photosynthesis. Being producers. So being able to take carbon, again, like carbon fixing, taking carbon out of the atmosphere and using it to make usable compounds for us. There's some estimates that say that protists, all the autotrophic protists, produce up to 30%, account for about 30% of the photosynthesis that happens in the world. So that's a pretty significant number. That 30% also means they're producing a lot of oxygen for us. And this is the foundation of food chains. Every organism in the world depends on photosynthesis for food, maybe directly, maybe indirectly, but this is the, again, foundational for the good of the world, the food chains of the world. Okay, so this is what we're going to talk about during class time if you come. I just want you to see this. Here are some suggestions for how to study for this test. Look over diagrams in the book and in the PowerPoints for topics that we covered, things like the trip operon, lac operon, how levels of eukaryotic gene regulation, the generalized virus life cycle we talked about, by both of them, the enveloped and the non-enveloped. Phylogenetic trees, very basic understanding of that. And some of the protist life cycles, again, the ones that we focused on in the PowerPoint are the ones you should look at. Chapter summaries are really good. There's some good tables, especially at the end of chapter 28. There are questions at the end of the chapter, multiple choice questions that you can practice with. Make flashcards. Study alone if it works for you. Study in groups if it works for you. Use your outlines. You can study your completed ones. You can take a blank outline and fill it in. There's lots of different ways, and again, we'll talk more about that in lecture. In the class meeting, the Zoom. A couple of things that I will say in terms of format. Right now, the test looks like it's almost done. We'll have about 20 multiple choice questions, and ranging in level of difficulty. Some of them are instantly, you should know them. Some of them you might have to think about. And then there are three or four diagrams to look at, and then a couple short answers. So you're going to need to use your time efficiently. You're going to need to be ready and go through it. And we'll talk in the Zoom meeting about some test taking strategies that I think are helpful. Study, 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 and I will see you on Monday. And I will figure out how to stop recording.